go. So there's this seminar coming up on Friday and next Friday, biotechnology. If you're interested in those topics, and you're interested in the RU Institute, these folks are from the RU Institute and they work here at USM. And here's this awesome kickball game coming up. That starts right after, or not right after, but soon after our last class meeting. And then this is what we're gonna do today, this week. So working on your term paper. When you're done with it, send me an email so that I know you're done with it. And I've already gotten a bunch of emails, and I'm in the process of responding to them. It'll take me quite a while to grade these because I have a lot of stuff going on. And then remember, we've got that answer key for a video 2, but get ready for the final. You want to look back at both the answer keys from exam 1 and 2. We're not going to have the same questions, but it could happen that I put a couple of the same questions on. But they're related, related topics. And then that's our final exam. It's 2 weeks from today. Here, it actually starts at 8, but don't get anxious if you're running a little late. Just come in quietly and get it started because the exam is going to be the same format as the first 2 exams, so you'll have plenty of time to get it done. You won't be pressured for time. And we'll loop back to questions for that, but now we're going to hear Sam's last presentation about CAR T-cell therapy.
Thanks, Dave. Good morning, everybody. How's it going?
Great.
Great. Thanks, Dave. Fourth talk today. We've talked about genes, we've talked about cell signaling, and we've talked about coordinated cellular responses. And today we're really going to talk a little bit about patients. So actually starting to think about what does it mean to give people CAR T-cell therapy? And then a few of the things that can go wrong with CAR T-cell therapy. Great. So just a few quick reminders, right? We're going to take these T cells out of patients. We're going to isolate them. We're going to use, we haven't talked about this, but it's one of my favorite parts of this story. We're actually going to use a lentil virus vector. We're going to use a retrovirus to insert the gene into the T cells. And that's, we'll talk, that is significant later on. So, and then we're going to culture them in little petri dishes in the lab, right? So this is all in incubators, people in white coats with pipettes, and then we're going to put them back into the patient. It's a chimeric antigen receptor, so we're combining the coding regions of several different previously existing proteins, combining those domains together to create a novel protein that is made from things that exist in nature, but does not exist in nature on its own. And this novel receptor activates multiple different pathways all in one. So T cells kill things, and we are taking all of the safety switches off of the T cells that go into your body. So they bind once and they kill. And we have, that's funky. They're supposed to be the same size. So genomic responses, right, we have these pathways that we just talked about, or that we glossed over. And now that we've been in this course long enough, we're going to recognize these. They lead to proliferation, cytokine production, and are anti-apoptotic. And we're also going to have non-genomic responses. And so this is the formation of this immunological synapse where it wraps around the releasing of the cytokines which recruit further immune cells, and then also cytotoxic factors that kill the target cell. And in the case of CAR T cells, that's a specific type of cancer. Okay, and I liked this one. I kept this one in, right? AP1, we've talked about that. Induced by co-stimulation, that's the multiple pathways in normal T cells, required for chromatin opening, right? So we're actually going to be remodeling chromatin during T cell activation. So CAR T cell is indicated currently relapsed refractory blood cancers. And this gets really, really complicated really, really fast because there are so many types of blood and so many different things can go wrong with them. But when we say relapsed refractory, what we mean is that this is for people whose cancer is not responding to other treatments right now. This is not just I just got diagnosed, we're going to go straight to CAR T-cell. This is currently one of the last treatments that people are being given. And that's for a few reasons. One is that it's so new. Two is that it's so expensive. Before we talk about this, so blah, 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 blah. Why is this here? Sorry. CAR T-cell, one of the reasons why CAR T-cell is a last line is that it is very dangerous to give. It has many, patients very frequently respond with life-threatening side effects that require extensive management, usually in an inpatient, so fully hospitalized setting. The first is called cytokine release syndrome. You might have heard this called a cytokine storm. And this leads to a hypotensive emergency, right, where your blood pressure drops so far and you aren't perfusing your tissues, and untreated, this can kill you. And this is a positive feedback loop where your immune system is releasing so many cytokines that you're falling apart. And we don't really understand this that well yet.
You normally have multiple limb amputations.
Yeah, it can be bad. It's not fun. Wow, the fonts came through. Sorry, everyone. This says CAR T-cell related encephalopathy syndrome. So it causes acute neurological damage and inflammation of the brain. And they're not exactly sure why, but this can also kill you, right? Your brain is filling with fluid, seizures, damage to various crucial organs. It needs to be managed really intensely. B T-cell aplasia and infections. So it can lead to like long-lasting reductions in your blood cell counts. So you can have long-term difficulty fighting off infections. And this is on top of you've already gone through probably years of cancer treatments, so your immune system is not in great shape to begin with. So you can really struggle with secondary infections and these are especially risky within the first month of treatment, but that's neither here nor there. Low blood levels, in addition, you can have not enough, you can have anemia, and that anemia specifically refers to red blood cells, but the corresponding effects for white blood cells and for platelets as well. You can just not be producing enough blood. And you can have what are called coagulopathies, so disorders of clotting, and you can have both excessive clotting and not enough clotting. So that can cause pulmonary emboli and other embolotic disorders, but you can also have problems with hemorrhages, and in some cases spontaneous. So this is all to say that like CAR T-cell is amazing. I wasn't able to find like really good numbers on this stuff because it's really complicated and we don't know, and they quantify it for like every single different type of cancer that exists. But the short of it is that some patients, there are enough people who have whose lives have gone back to normal, is probably the best way to say, and who are just responding at 6 years are still cancer-free, right? So like kind of like truly incredible things that no one thought was possible before this, right? So we're seeing these like huge responses and also major side effects, problems to give, and it's a broad range, right? Some patients just don't respond at all. It's very common for this to relapse after about a year, but also we are seeing people just kind of like totally off the charts on bell curves. And there's a lot of questions about why that is and what's different between these patients and what's going on here. So very much a site of very active research and investigation is going on here. So relapse does frequently occur in CAR T-cell therapy. And I mentioned, and each cancer is different, and we've talked about this kind of like, we can target a specific protein on the cancer. And which protein we're going to target depends on which type of cancer we're dealing with. And what we're seeing is different responses based on the different proteins that are being targeted. So relapse occurs. Antigen escape is the most common one. So cancer proliferation is driven by natural selection. And we've just created this cell that's going to kill every single one of the cancer cells that expresses a certain protein. And any that have, but also cancer cells tend to have the ability to mutate rapidly. So any of them that don't express that antigen will be selected for. And so you can see what it's frequently seen as a loss of the antigen in question on the cancer, and the cancer comes back now without the protein that you were binding to. CAR T cell persistence, this is a struggle in the long term. Like, how do we keep the CAR T cells in your body for long enough to be able to keep fighting the cancer? And this is where the co-stimulatory domains come in that we've talked about, right, that induce proliferation and anti-apoptosis. And also combining them with a drug seems to have, so you're both using chemotherapy and cellular therapy as part of your CAR T cell is really important. And then the final thing is the tumor microenvironment. And this is really the kind of answer that researchers have settled on. So we've mentioned at some points that CAR T cell therapy only really works for blood cancers. And the question of like, why is that true has really been is one of the major driving questions of oncology research in the last decade. And the answer they've come up with is something called the tumor microenvironment, which is the idea, and we're gonna really focus in on this one as the like conversation for where we go from where we keep going on this talk is, because it's really wild. So this is a picture of a solid tumor. There's a lot going on here. What are the big takeaways? The big takeaways are that tumors are actually heterologous and are composed of many in addition to cancer cells, they have recruited many non-cancer cells to this location and to this area. And that these tumors tend to have immunosuppressive tendencies. And and you can go get a PhD in this. But today, we're going to talk about the PD-1, PD-L1 pathway. So this is a very common way that the tumor microenvironment suppresses the immune response. PD-1, programmed cell death protein 1, programmed cell death ligand 1. And so this is, if you can see it in the bottom right here, this is our cancer cell, and it's expressing the ligand on its cell surface. So this is a transmembrane protein. And this is a T cell, and it expresses the protein. So the ligand binds to the receptor and triggers an intracellular response. I was really hoping these would be more visible. I'm sorry, everyone. Okay, let's see. I think this next one, too complicated. Okay, this PD-L1 pathway in T cells suppresses activity, decreases proliferation, decreases cytokine secretion, and decreases T cell survival. Right? So what we're seeing is that one of the ways that the tumor microenvironment fights off the immune system is by expressing PD-1, which binds to PD-L1 on T cells, decreasing their activity.
So they're causing T cells to undergo apoptosis, basically.
Or I would probably say it's more accurate to say increasing apoptotic genes. The language in this that they like to use, that the literature likes to use, is exhaustion. And so I don't know exactly how they quantify that or what that means. But my understanding of that is you have this population of T cells that are active at lower rates than you would otherwise expect.
So it's inhibiting a lot of the genomic responses, not really much of the non-genomic.
But also cytokine secretion, right? So we are also inhibiting non-genomic responses.
Okay.
Yeah, right. So yeah, we're increasing a lot of those activation responses and those direct killing responses. Super good question.
I I think you're saying right, because like they can increase their cytokines, which is a genomic response, but the actual release is just a non-genomic. That's how that works.
Yeah, and kind of like Dave's conversation about like effector versus regulatory proteins, like at some level it's useful, and at some level if you push it far enough it breaks down. Okay, so this is kind of showing this again. In the bottom right, there are, you also see, yeah, I think let's talk about this. So we've talked about how in T cells, activation of these pathways leads to a coherent response. Activation of pathways across different cell types also tends to lead to a coherent cellular response. So in this situation, PD-L1 binding to PD-1 on Treg cells, and those are T regulatory cells, suppresses the immune system, and that's their job, right? So what we're doing is this PD-1, PD-L1 pathway just turns off the immune system, right? It just turns it down, and it does that through activating Treg cells. And also, if we look here, right, here's so many of our friends. Here's the RAS-MEK-ERK pathway that's being suppressed. Here's PTEN that's being suppressed. Here's PI3 kinase, which is being suppressed, right? All of this through this PD-L1, PD-1 interaction. And so the big takeaway here is that tumor cells evade the immune system and evade CAR T cells through the PD-1, PD-L1 inhibitory signaling pathway. Okay, so now let's talk. So we've talked about CAR T cell therapy, right? Things going on with it. It's really dangerous to give. It takes a really, really skilled care team to give it. While there are these incredible off-the-curve responses, it is associated with relapse after a certain amount of time. We're most interested in relapse driven by the tumor microenvironment. And there's just a logistics considerations that need to go into giving of CAR T cell therapy. It's really expensive. Costs up to $1 million per patient, right? And so because it's highly specialized, you are like literally taking one patient's cells out, growing them in a lab, transfecting them with a virus that you have to be culturing and maintaining in a lab. That virus is dangerous. You have to put them back in.
And I'm assuming insurances assume it's too experimental.
I don't know the current state on insurance, but yeah, insurance-based medicine doesn't always lead to good patient outcomes. That's definitely true. And there has been some question about whether CAR T cell can cause secondary cancers. And there is one, this guy, like Dave talks about this, right? These like cancer stories where it's just the worst luck in the world. And there is one confirmed case where the random insertion of the CAR T cell receptor was just in a gene that caused a secondary cancer in blood cells. But overall, the current perspective is that the CAR T cell is probably not driving secondary cancers. But that's still being researched and is something that people have been worried about in the past. Future directions, they're thinking about including 2 targets. Down here in the bottom is kind of a weird little eyeball symbol, suggesting all the different possible targets that they might try and use for CAR T cell that they have identified and are working with. And I talked about how there's this, once you induce CAR T cell, there's a natural selection pressure for the cancer cells to no longer express the targeted protein. So they're thinking about using bispecific T cells or T cells that can target 2 different proteins that would be found on the cancer in order to prevent that escape. I mentioned coupling with chemotherapies. They're especially interested in inhibiting that PD-L1, PD-1 pathway because that will prevent the CAR T cells from being inactivated by the tumor microenvironment. And yeah, expanding the protein targets, right? We have this huge list of possible proteins for a variety of different blood cancers that they are interested in targeting. So that's a little, this is the slide where I'm supposed to show you like some photo of like a successful cancer story, and I just couldn't find anyone that like didn't make me sick in my stomach, because I think there's like these photos are like all selected for like these 7-year-old kids who come from like obviously like upper middle class families, and I just think it really like is like a moment to say like there are real stories of people out there who are like alive today who would not otherwise be because of CAR T-cell. And let's not play the game of who deserves healthcare and who doesn't. And does anyone have any questions?
For a patient who's getting CAR T-cell therapy, do you have to be like in ICU for that level monitoring, or can you get it on a regular like medical?
Yeah, that's a good question. It's going to depend. I don't actually know like what this is doing. I don't know if anyone saw it mentioned. Maine Med is now giving CAR T as of last year. So this is an expanding treatment, but to give that like some perspective, right? Like Maine Medical Center is a level 1 trauma center, and they just started offering this treatment last year, right? This is like really new. So I bet you could go talk to the oncologist there, and they would probably be like, we're still figuring that out. Yeah. All right. Thanks, everyone. Thanks to Dave and Riley.
You know what? We need to find it.
Yeah.
And here's a few papers you could go read if you really want to go
Which
wild.
one is the one about the patient who had like the unfortunate?
It's
sounds
this.
interesting.
It's the fourth one. Osdemeril M. That one right there. Indolent CD4 plus CAR T cell. That was published in New England Journal of Medicine last year. Thanks,
Poor
Vanna
guy
White.
went to go get cancer treatment and then
Guy
he
got
gave himself
cancer.
one of those
Yep.
rats.
Yeah, I know.
Contusions.
We're not supposed to laugh at that. It's bad form. It's a very short write up. It's like a paragraph.
All right, super. Thank you, Sam. And so remember, the purpose of having Sam participate is to help you build different kinds of learning contexts. So you can think, oh, is that guy in class that stood up and told us about CAR T cells? And then your brain comes to that place. And that's just a big part of my goal of this course, is to think about learning, and learning is all about recall. And when we recall things, like a lot of times people will say, oh, there was this really traumatic thing that happened, and I remember right where I was when it happened, right? Because it's context dependent, and you can use the context to help your recall, so you can build that and study in different locations and so on and so forth. So great. And so on we go. And so this is where we're at, and that is we just covered one chapter, the nucleus, in chapter 10. I'm actually done talking about the nucleus chapter, and we're going to roll on. You don't need to read any more parts of your book, and we're going to roll on to just more molecular biology and lace the course up, because we have today and then 3 more class meetings, and this will be new stuff on our final exam. Our final exam will cover everything. There'll be not a particularly strong focus on this material, but definitely it's new. And you'll see that we have a very logical way to just keep adding stuff, even though you aren't expected to read the book after chapter 10. So in chapter 10, there were these 3 pieces, and one was the nucleus and the nuclear membrane, and especially the nuclear pore, stuff getting in and out. And that matched up really well with the topics that we've learned about in our course, because we've learned about so many proteins that need to get into the nucleus to function. And so that opened up the idea of, after a protein is made, we've seen lots of specific examples of how the protein is regulated, and that's called post-translational regulation. And one of them that we've talked about since the start of the course is, if it's a transcription factor, can the protein get in the nucleus? Can FOXO get in the nucleus? And so that's a good example of how we saw a simple post-translational modification, phosphorylation, and that regulates protein's activity. And so that's part of the story of regulation of gene expression or gene activity, is, well, you could turn that activity off at the level of the protein just by phosphorylating it. And then we add to it that for FOXO, that phosphorylation turns it off. And it turns it off by preventing it getting to the nucleus like that. And 1433 simply covers up the nuclear localization signal. FOXO can't get in. So really nice way of adding to that knowledge we started our course with. here by saying, in a cellular context, a better understanding of that entry and exit from the nucleus. And then we just had brief things to say about these other two parts. So the chromosomes are organized in their interphase nucleus, and we see heterochromatin and euchromatin distributed unevenly like that. And so we talked a little bit about that, but there's just a very little bit of stuff in there. And then the same thing with nuclear bodies. We learned about some nuclear bodies. We learned that the euchromatin is localized in these transcription factories. So, speckled spots in the nucleus have a lot more transcription than other regions. And the same thing is true in S phase. The DNA replication factories are localized within nucleus. And we learned about the nucleolus. It's the site of ribosomal RNA synthesis and assembly of the ribosomal subunits, like that. So, and we learned a little bit about polycomb, and polycomb is a repressor of gene expression. So there are polycomb bodies, or sort of localized region of the nucleus. So again, the interphase nucleus is just a gray haze, but there's localized activity in it. And that's all relatively new stuff, and it opens the door for regulation of those, like entry into those locations and so forth. But that's still fairly new stuff, and people are learning more about it. So that's the deal with chapter 10, and we can pause here and ask any questions you have about anything at all, because now all these class meetings are also, study time, review time for the exam. I don't review, but we can review as much as you want by asking questions. Yes?
When we talk about the polycomb proteins, I was just a little interested on how they did the whole group shutdown. I just didn't know if that was because the genes are so wound up that there'd be multiple genes, that then like when they phosphorylate that histone, it shuts everything, or not phosphorylate, I'm not sure exactly how they do it actually.
Yeah, so we learned some about the idea that, especially with the estrogen receptor, that there's this region of the chromatin in the region of the gene that is being regulated. And in the case of the estrogen receptor, it's because it's tethered there. And polycomb binds to those histones, binds to the same histones that the estrogen receptor can modify. And the polycomb protein can repress. So it's a way of closing down chromatin, and the polycomb has to find it. And people are really excited about polycomb, and there's a lot of work done on it. But the bottom line is, fundamentally, it has to be recruited to a region. And it's traditional transcription factors that do a lot of that recruiting. And people don't talk about that very much, but again, something like the estrogen receptor, not the estrogen receptor, but something like a transcription factor can actually help recruit polycomb. And because it's tethered, it's recruiting polycomb to this whole region of area. And the polycomb is just going to cause the chromatin to get tighter and tighter, and it's always going to be repressing gene expression.
Because in the photo, it looks like little brains almost sitting on top of them, and I didn't.
Birthday parties, yeah, it looks like little birthday parties, right?
I just didn't know if that was the whole protein itself, and that just they come together. It just seemed dangerous to shut down a whole region.
Yeah, and it is a kind of a concern, but it turns out that genes are really spread out. There's people don't talk about this very much, but there's just huge gaps between many genes.
Like the junk DNA.
Yeah, just huge chunks of space that they're chained together by DNA sequence, but they're just far apart. Yep.
And can these be removed? These aren't like the constricative. These are the facultative, so you can remove it.
Yeah. Although polycomb often is a permanent thing where it's going to be permanently done.
So then it wouldn't be on estrogen because you would need to open estrogen.
No, yeah.
Okay.
Yep.
You just talked about how genes are very frequently very far apart within the chromosomes, but also I've seen in some places the idea that you can have multiple genes on top of each other just with a different reference reading frame. How does how do we fit these two pieces of knowledge together?
Yep. So there are a few genes that are close to each other because they're co-regulated. Sometimes students have heard of genes called HOX genes, H-O-X, and Hox genes are clustered together because they're co-regulated. So for that to happen, oops, I forgot to get my little drawing pen out. For what it's like is over evolutionary time, there's a lot of these chromosomal breaks and rearrangements that occur over long periods of time. And so in general, here's a good example. In general, when we see a gene family of paralogs, right, they arose by gene duplication. They must have been neighbors. When we see gene families, they're scattered throughout the chromosomes, throughout the genome. And that's because over evolutionary time, there becomes, although it's very rare, there becomes a lot of rearrangement of the chromosomes. And so we see that where we rarely see a gene family all clustered together. The Hox gene family is one of them, where they are said to be collinear, they're all close together. There must be some evolutionary pressure to keep those clustered together. And so one way to think about that is that, yeah, sometimes the genes are close together, and sometimes there is natural selection pressure for maintaining them because they're co-regulated in some way, but it's really pretty uncommon. Like I said, of all the gene families, I only know one where the gene family has stayed together over evolutionary time. Like Hox genes are found in all animals that have anterior, posterior ends, almost all animals, and they are always together. And that's because they're regulated together in this coordinated way. Yep. Other questions people have about anything at all? And again, we could loop back to, the exam answer key videos, right? You can have questions like that, or pages in the book, things like that. Yeah.
We use the word recruited a lot of the time, and it just feels like, I'm wondering if we can dig into that word. It feels a little like hand wavy to me, like.
Recruited, yeah.
It's chemical messengers, right?
I mean, it must, yeah, like,
Well, like, what do we, like, cause like, it's not like the protein's walking around, like, giving out leaflets being like, hey, I'm throwing a party, come to my party, right? Like, it's not a thing that.
Yeah, so, let's think of a good example. So, I'm gonna skip ahead some slides, just to some slides we have that we'll cover by the end of the semester. So, just to map out, where we're going. So where we're going to go is, for the rest of the course, is we saw last week that although we've emphasized transcription regulation in this course, that's just the first step in gene expression. First, we've got to transcribe it. We get a primary transcript. And then within the nucleus, there's going to be this RNA processing to go from a precursor primary transcript to a mature RNA that gets to leave the nucleus. So we're going to talk a little bit more about that RNA processing, learn a little bit more about that. Then, if it's a coding gene, it leaves as a messenger RNA, binds to the ribosome, gets translated, and then there's this post-translational regulation that includes phosphorylation and so forth like that. And we'll talk a little bit about that. And so that's that from introbiology, the actual full pathway of gene expression. And in talking about it, we're going to spend some time talking about this post-translational regulation, okay? And so to answer your question, we'll go to a specific example that'll be a new one for us, and it's fine. And the example is, one example of post-translational regulation is the protein gets destroyed. So how long does the protein last? And we've seen some examples that we just took in stride, right? MYC protein gets made, it does not last long. It lasts about 5 minutes because it's just so dangerous. So we saw MYC is unstable. We saw beta-catenin, that's a transcription factor we learned about, that is activated by the Wnt signaling pathway. In the absence of the Wnt signal, beta-catenin is just constantly being destroyed. Then another example, and we talked about that's in our book, is that p53 protein, that transcription factor. We especially emphasize when it gets phosphorylated, it's going to bind to the enhancer of the p21 gene and activate the p21 gene expression and stop the cell cycle. Our book showed and talked about it before it gets phosphorylated, P53 is just constantly getting degraded. So those, and maybe we have another example, but those are three good examples of this idea of post-translational regulation by degradation. And the protein is going to get destroyed by proteolysis, by proteases. And remember, we talked a little bit about those, how there's some specific examples of protein cleaving, like how caspases clip proteins to activate them, like the scramblase. Then there are other proteases that just destroy proteins. And we usually think of our own digestive system, where we're just going to digest proteins and break them down to amino acids, but that's present in each one of our cells, is this protein degradation. It turns out that when a protein is going to get degraded, beta-catenin, P53, MYC, all of them, there's a way that it usually gets destroyed, way it gets degraded. And so we're just going to skip to some slides that's new to get to that question, okay? So I'm just going to find them. Yeah, I know, it looks like we're going to cover a lot more stuff, doesn't it? So here's just a starting picture of this, where it's post-translational regulation, and it's in muscle, so we like muscle, so it could be in there. And this just shows proteins, and it shows a variety of ways that they can get modified. And one of them is they could get phosphorylated, so we're superstar experts on that. That's a post-translational modification. It's a covalent modification. A modification occurs as an enzymatic reaction catalyzed by a kinase, a specific kinase, and then it could be removed by a phosphatase, a specific. And so that's just one. And we're going to learn about another one, and we're going to learn about it to answer this question, and it's over here, and it's a way that we tag this protein to get it destroyed. So it's called, and it's over here, and you can see there's a protein, and instead of having a phosphate group added, there's a little protein, just a short protein that's added, and the protein's called ubiquitin. So we're going to ubiquitinate it, not phosphorylate it, we're going to ubiquitinate it. And so here's the protein, it's ubiquitin, and it's just small, like 10,000 Daltons. So it's a protein, but it's a small one. And we can add one, and then we can add more. And we're tagging the protein for destruction. So when we ubiquitinate the protein, usually what we're doing is we're saying, we got to get rid of this protein. So it's just a tag, right? Okay. So it says ubiquitin plays a crucial role in protein degradation, acting as a tag that marks proteins for destruction by something called a proteasome. And a proteasome is a curious organelle that you probably never heard about before. It's not very big. It's about as big as a ribosome, and it looks like a barrel, literally a barrel, like a cylinder, like a trash can. It really looks like a trash can. And proteins go into the proteasome if they're tagged with this ubiquitin. And it usually takes multiple ubiquitins to get them in there, so they're called polyubiquitinated. So we're going to tag it, add a covalent bonded ubiquitin, and that's how you get into a proteasome. And I'll just show you a slide. There's a slide. So here's a protein, and it can get ubiquitinated, and then likely more ubiquitin, and then it's going to go into this trash can called the proteasome. So those are two terms we want to learn for the final. The proteasome is a little organelle that's like a trash can for proteins. And proteins usually get in there. Main way of getting into the trash is they get tagged, okay? So this would be the example, your question would be, one example is ubiquitinating proteins recruits them for entry into the proteasome. So the proteasome has a little, like the trash can has a little lid, and it opens if a protein's been ubiquitinated. So that's one way we use the word recruit. And so it's a little bit of maybe of a flawed thing, you know, because like, think about it, where it's almost like, you know, we have the license plates on our car, and you're supposed to have that tag for the correct year. And if you don't have the right tag, you're being recruited to being pulled over by the cops. It doesn't feel like you're being recruited to get pulled over. That's just the term that's used. Another way to say it is we would maybe tag it, you know, and it's not a, you know, it's an okay way of using recruited, but it's kind of a little bit of a curious use of it. There's other context, right, that we would say the word recruit. Yeah, and we could talk more about that. But this is an example where people would say that ubiquitinating a protein recruits it for entry into the proteasome. So see, and that's, I think, a really common way, and I would call it tagging it, you know, like that. So there are other contexts too, though, and you know, there would be a variety of examples of that to answer that question about what does it mean to recruit something. This would be one broad example, is literally to tag it, and you're recruiting it, you know, like that. You're like marking it for entry into the proteasome. Yep.
Would it be wrong to think of this like protein program cell death? Because I see that you're breaking up into its amino acids, so then that could be recycled. Like I know that it goes and then it can enter once it's tagged, but like it's been chosen for death. It then goes, it gets chopped up, like.
Yeah, so there's two big famous ways of destroying proteins in a cell. One is the proteasome, and the other is a lysosome. So the lysosome is a famous organelle that's got an acidic compartment to it, and it, like people call it the stomach of the eukaryotic cell because that is an acidic compartment, and that can just hydrolyze the proteins down to amino acids. And so when we talk about apoptosis and autophagy, the autophagosome works by engulfing things inside the cell and then fusing with lysosome. So they're not going to be destroyed by the proteasome, they're getting destroyed in a lysosomal type
And do
stomach.
those proteins get tagged for that?
Nope, it's just engulfing big chunks of areas. Like all the whole mitochondria, all the proteins are gonna get destroyed, not by a proteasome. So again, there's two famous ways of degrading proteins, and one of them is this proteasome, and the other is the lysosome. That's true for apoptosis too. The big destroyer of proteins in apoptosis is when the other cell phagocytizes the dying cell. Because when it phagocytizes it, it brings it inside and it fuses the dying or dead cell with its lysosomes, and it turns it into kind of like a stomach digestion thing, where it's really the low pH that facilitates what's called acid hydrolysis of the amino acids, the peptide bond. Yep. But this is proteasome and degradation, like that. So that's just to jump to that. And it's an example of post-translational regulation. And it's how these proteins get destroyed. So just a little entry into that example. Ubiquitination, it can do more than just tag or recruit protein for degradation, but that's its most famous role. And so nice visual there. And there's just lots of these little proteasomes throughout the cell. And literally, I didn't think of it before, but I mean, it's a perfect analogy. It's a trash can for proteins. And usually you have to, the main way to get into the trash can is get ubiquitinated, and then you're gonna end up in the trash can. Yeah? So for all three of those famous examples, so beta-catenin, P53, and MYC, they also have an additional mechanism. So I'm just gonna show you that because it ends up that this post-translational regulation gets complicated really fast. So we'll keep it simple, and we'll say, yep, on the exam, I'll say, give me 3 examples of proteins that get turned over fast because they get ubiquitinated. And you'll be like P53, MYC, and beta-catenin, right? That's the main way to think about it. So I just show you this just so you can see this, and we'll just read it in a little bit. So ubiquitin plays a crucial role in protein degradation, acting as a tag that marks proteins or recruits proteins for destruction by the proteasome. Proteins targeted for degradation are attached to ubiquitin molecules. They're a covalent bond. An enzyme adds it. Protein is tagged with multiple ubiquitins. It's recognized by the proteasome and enters the proteasome, and proteins go in and they don't come out. Yep. Then it says, well, the ubiquitin proteasome system is the primary role for protein degradation in eukaryotic cells. Proteins can also be degraded by the proteasome without the need for ubiquitin tags. So, it just gets expanded, and we don't have to learn that. Just the awareness that there are additional pathways, but that's the big one. the main one, like that. So there are other pathways, and so let's just see this here. So it says ubiquitin dependent degradation. The classic pathway for CMYC degradation involves phosphorylation of CMYC, and that is then recognized by an enzyme that ubiquitinates it. So again, you don't need to learn this. Well, you do need to learn this, just that MYC is going to get ubiquitinated, it's going to get degraded right away. It's like, we don't want you around here, right? And so getting destruction. Well, ubiquitination is a crucial step in the degradation of many proteins, including MYC. Ubiquitin independent pathway mediated by the AZ2 and midnolin demonstrates that proteasomal degradation can also occur without this ubiquitination step. So don't worry about that. Don't worry those proteins, just the idea in your head. It's more stuff than this. And when we look at these special proteins that are like really unstable, they often have both the ubiquitinated proteasomal destruction and they have another mechanism. Why do people care about that? Well, because sometimes these proteins like AZ2 and midnolin are issues in cancer because their MYC's not getting this, not getting degraded, right? So those could be considered a little bit like tumor suppressors. And if you screw those up, you have MYC around too long, right? So that's why, people be like, we got to talk more about it. But we don't, because, we got a final exam in 2 weeks. So again, primarily, MYC get destroyed by ubiquitinating it and degrading it, just right away, okay? Then beta-catenin. Beta-catenin degradation is often linked to ubiquitination and proteasome-mediated destruction. But studies have shown that degradation can also occur through alternative mechanisms. Some studies suggest that beta-catenin degradation can be achieved without ubiquitination, potentially involving mechanisms of other kinds of modification and so forth. Again, don't worry about that part. Primarily, it's ubiquitination and proteasome destruction. P53 degradation is primarily regulated by the ubiquitin proteasome system. Some mechanisms exist where P53 can be degraded without ubiquitination. Here's this protein, MDM2. MDM2, a key regulator P53, can interact with P53 in a ubiquitin-independent manner, promoting P53's association with the proteasome for degradation, even without ubiquitination. So see, there are these other pathways of getting a protein into the trash, but they're not as important as the ubiquitin one. So we learned about the ubiquitin and just an awareness that, especially for really highly unstable proteins, there's probably another mechanism for recruiting them to the proteasome. Yep.
So for that, you just want us to basically know that like MYC, beta-catenin, and P53 are often destroyed via ubiquitination.
Yeah, because that's a great example, right? Because we're going to talk some in this week and next week about the pathway of regulating gene expression, and we're going to get to examples of post-translational regulation. We've made the protein that the gene is fully expressed. No, because we know it could be phosphorylated. That might be necessary to turn it off or on. Now we learned another one. It could get ubiquitinated and destroyed. And that's the main way that MYC, beta-catenin, and P53 all get destroyed. And we just opened a crack to realize, for your summer reading, you could be like, I'm going to go to the beach with some really excellent science articles about other ways to get into the proteasome trash can instead of ubiquitination. But we're not going to learn about that for the final. Yeah? So it goes back to this image where in post-translational regulation of gene expression, we learned about phosphorylation and ubiquitination. We didn't learn about it before. It's involved especially in degradation by entering into the proteasome. Yes and yes.
Where's this image from?
Let's see if I have it. It's from some review.
Okay.
It's obviously not from your book, and it's from a review. Let's see, that I hope to have some slides because we could go back to some muscle stuff. So let me just show you that a little bit. So we'll just jump to this and deal with this too right now, okay? So I said, we've learned a lot about muscle differentiation, and what we learned about is that we're gonna activate gene expression. So that's the front end. Muscle differentiation is gonna activate gene expression. And that is the big story, transcription activation. And that's generally the story in development. So development is how we form and grow. So we're gonna take some cells and we're gonna make muscle cells. The cells always have all the genes, genomic equivalents. So to become a muscle cell, probably gonna involve new gene expression. And the most logical thing is those genes are not being transcribed, and now they're gonna get transcribed. And that's true for muscles and every other organ in our body, bone, liver, brain. We got all the genes. Probably the most important first step of becoming a pancreas cell is to express some new genes, turn on the transcription of new genes like that, right? And so that's a great place to start the story, at the first step, right? And so we talked and started our course with this, and this is the pathway that we've learned about, where the cells are proliferating, and they could still be satellite stem cells or early stem cells, and they're gonna start to differentiate. So they're myoblasts, and they activate MyoD, and they stop the cell cycle, and they express myogenin, and they make muscles, so they make muscle proteins. And you don't have to learn more from this. I just want to show you that there's MyoD, but it's showing a lot of other things interacting with it. And some of them are familiar, like NF-kappa B and these SMADs and stuff like that, and some are not. There was at the start of our course, some people always ask, well, how does MyoD get activated? And this part starts to talk about other transcription factors that are going to activate MyoD, and it's a complicated story. We just focused on, of all the dominoes falling in a line, MyoD, myogenin, and muscle genes, because it's great, because it's pretty simple. But you can dive into it more, and you can see there are more players. And then we're going to turn off the cell cycle. It has some familiar factors like retinoblastoma and stuff like that in there, but other things going on too. So I just wanted to show you that, toward the end of our course, right? That we have a really good framework, again, to learn more about muscle differentiation, and there's more pieces to it. Here's this transcription factor, MIF5. MIF5 is just as cool as MyoD. What does that mean? If you have a mutation, you have a mouse that has a mutation and destroys the MyoD gene, there's no muscle. On the other hand, we learned if you force the expression of normal MyoD in a precursor cell, they'll become muscle. So we can say that MyoD is necessary to make muscle, and it's sufficient to make muscle. And the same is true for MIF5. MIF5 is another transcription factor that is just as cool as MyoD. But we're not going to go there. We've got enough stuff that we learned about the transcription regulation and the differentiation of muscles. So just to say that, that's the first step in gene expression. And then we're just going to use this because it turns out that there's more to muscles than we've learned about in our course. Another thing that's really famous about muscles is when a person gets old, their muscles start to disappear. Sometimes because of disease, sometimes of inactivity, but also just by age. An older person just starts to have fewer muscles, like that, okay? So that's a different story than we learned about. And it turns out that there's a transcription story there that's really interesting, but there's also a story about the RNA processing and a story about post-translational modification that actually become a bigger part. So we started our course learning about making muscles as a young person and not losing muscles as an old person, because making muscles as a young person is complicated, but it's really focused on transcription activation. And there are some stories of later steps of RNA processing and post-translational regulation, but it's really emphasizing transcription. And again, that's logically just because you got all the genes and you want to make muscles, you're going to have to turn on those genes. So that's a big part of the story. On the other hand, in this old person, there's a story of transcription regulation, and we're just going to take a peek at that just because you're like, oh my gosh, we can understand some of that from our course. It's at the other end of the life cycle, right? But we can understand some of that stuff that's going on. And then it turns out that it's partly about transcription regulation, but it's got a lot of stories about that RNA processing and the protein post-translational regulation. So, you know, it becomes a story that's kind of a more advanced story. But again, it's just natural aging. Like that, okay? So let's just look at that a little bit. So here's a hormone called myostatin, and it's going to bind to a receptor, and it's going to cause people to get old with their muscles. It's going to decrease muscles. And I'll just skip to receptor figure. So when we looked at our chapter on chapter 16 on receptors and signal transduction pathways, we learned about receptor tyrosine kinases. We learned about gene-protein coupled receptors. Then at the end of that chapter, we learned about some other receptors, like the Wnt signaling pathway and beta-catenin we learned about, a little bit about Notch, and we learned a little bit about this one, and we learned about it simply because this one activates a transcription factor family called SMADs. When they get phosphorylated, they activate transcription. And myostatin is in this family. So there's a ligand signaling molecule called myostatin. It's a hormone, and it binds to a receptor that works this way. And myostatin is going to activate transcription by activating these SMADs. So this is an example of myostatin, and it's involved in a person getting old and making less muscle, like that, and having started to get weaker muscles. And you know, they can work out more, and they're just not going to build their muscles as much. I can feel that right now in my life. I'm old, and I can feel like when I exercise, I don't get like in better shape as much as I used to. I used to exercise and I'd do it like one time, and I'd be like, I'm back in shape, you know, just like that. I mean, it's pretty nutty to grow old. And so it's that. Yeah, do you have a question, Alex?
Yeah, so is myostatin, is it a transcription factor?
No, it's outside the cell, it's a signaling
It's just
molecule.
a hormone?
Yep,
Okay.
yep, it's a signaling molecule outside the cell. It's going to bind to this receptor, and its action is going to be on muscle cells and satellite stem cells, and it's going to act partly by transcription, but also by other mechanisms. Okay, so because we know transcription well, let's just take a peek a little bit, right? So just to show you, back when we were making this list of transcription factors, we're like SMADs are, you know, very briefly, right? Probably there's a good chance you don't remember, but if they get phosphorylated, they're activated. So lots of transcription factors regulated by phosphorylation, okay? And so let's see if I have this in here right. So yeah, so here's an article. Sorry, it might be the same one you asked about, Sam, but I don't think so. And I'll find the one that you asked about, that other post-translational one. So let's just read this abstract, and then we'll have a little concept chat. So it says the transcriptional regulation of skeletal muscle development, myogenesis, has been documented for over 3 decades and served as a paradigm for tissue-specific cell type determination differentiation. That's what I've just been saying. It's a good example of how we get all the different tissues, brains and livers and so forth. It's just a good, satisfying example. Is we're going to turn on some new genes. We're going to activate transcription of new genes. Myogenic stem cells, so our satellite stem cells, they have a different kind of abbreviation, and adult satellite stem cells and so forth, regulated by transcription factors for their stem cell characteristics, while their lineage determined is determined, both dictated by the myogenic regulatory factors that compromise myogenin, myod, and these other ones we didn't need to learn about. Okay? So they're talking about the precursor cells, the myoblasts, and the stem cells, right? And so they say, yep, the myocyte enhancer factor MEF2C is activated by MRF during terminal differentiation, collaborates and so they're talking about what we learned about. Then they say recent studies have found critical regulation of these myogenic transcription factors at mRNA level 2. So there's more to it like that, including subcellular localization, stability, translation regulation. Therefore, the regulation of these things involves more than just transcription. Great. And so they're talking about the regulation of these after transcription. That's great. So we have that example of that, and that's part of the story we learned about, but a smaller part like that. And then, let's see if I want to go to this. No, I might just not have the right one in the right place. I don't think I have it. Yeah, so there's a couple of slides that I wanted to show you about that, more about myostatin, that I guess I might not have gotten in here at the right thing. Yeah, here they are. So it says, now this is going to be this myostatin, that's the older age one, right? So it's a good one, like, right, just like how we talked about, let's think about different places. Myostatin is an inhibitor of muscle development, and it especially plays a role late in life, okay? So it says myostatin negatively regulates satellite stem cell activation and self-renewal. So remember we learned about stem cells, and adult stem cells divide asymmetrically. The muscle satellite stem cells are adult stem cells, so they're dividing, and a stem cell divide will make another self-renewing stem cell and a precursor cell. So they'll do that anytime we need more muscles throughout our life. And then toward the end of our life, myostatin is going to be like, yeah, we're done with that. Get ready, to go, to be old. So it says satellite stem cells are, resting muscle stem cells that propose postnatal after birth muscle growth and repair. Here we show that myostatin, a TGF beta family member, so it's going to act that way, signals satellite stem cell quiescent and negatively regulates satellite stem cell renewal. Then it says down here, it says cell cycle analysis confirms that myostatin upregulates P21. A CDK inhibitor, and decrease the levels and activity of CDK2 protein in satellite stem cells. Myostatin regulates the G1 to S progression and thus maintains the quiescent or stopped status. So one thing myostatin does is it blocks the cell cycle by activating expression of this P21 that we know so much about. And I'm sorry, I mentioned we're going to have a concept chat, but let's just add to this. And again, we're still talking some about transcription regulation. And so this is that P21 gene, right? It codes for a P21 protein. And the P21 protein is a small inhibitory protein that blocks the cell cycle at start. And we learned that phosphorylated P53 transcription factor binds there. And it activates expression. And that was on our second exam. We drew a little picture of it because it's really important to appreciate that P53 does not bind to P21 protein. These two proteins never meet. P53 activates expression of that. And what we just added to here is the implication that there must be another enhancer of P21 that would bind to one of these phosphorylated SMAD proteins that gets activated by a receptor and a signal transduction pathway that is bound by that myostatin ligand. So the myostatin does that to a muscle satellite stem cell. So this example is just to say in the old age, there's still transcription regulation by myostatin. Still familiar stuff. We're like, we could jump on board that right away. We just did. We're like, okay, yeah, good. We're going to change gene expression like that by transcription regulation. We're going to have the transcription factors regulating and activate or repress, gene expression. And then there are these additional examples, like that there is that transcription regulation in that old age muscle wasting, but there's more examples of RNA processing and translation regulation, like that. So let's see, maybe we don't even have time. We do have time, so let's have a concept chat and talk about some things we learned today that were important that we learned about CAR T-cell therapy. So you might want to quickly partner with Sam or anyone else, because we heard what Sam had to say, or this other stuff we're talking about.
I like the ubiquitous being used as a verb, like, and it sounds like multiple combined, and then more combined, the more likely you are to go to the protease. And then that just means
I was confused about cortisone in cytoplasm or is in because all those
cytosol, because that's where the protein, I would assume. That's really good for cylinder. Like, so if you're ubiquinated, that is a protein that is binding to another protein, basically, right? That then marks it to go to the protease. I'm assuming that's. Assuming they do, because probably have some way of getting things. Or encapsulation.
they're putting actually you have like the safe zone in the enemy territory. Like all this stuff happens. Once you get to know those,
I'm either having a migraine right now, or I'm serious. Like, I have a brain fog. I've been around by and all of a sudden, my head hurts, and I'm seeing tons of my migraines. I have those shots. And I'm like, I feel like my handwriting got significantly worse now.
I'm so tired today.
I'm like, am I having a headache, or am I having or do I need to go to the ER? I am not going to the ER. My proteins will be great. I literally think they have 2 hours. So I can go after my lysomy. Yeah. But no. Okay, so then also we learned that MIG, beta-catenin, and I can't even read my handwriting. What was the third one? MIG, beta-catenin.
P53.
Can be destroyed by the ubiquitin? Was it P53?
Yep.
Also we learned that SMADs can activate the P21 protein.
Yeah, that was a full circle thing. It was like a side note earlier, now it's like, okay, now they're important, so. Yeah. Go back into this.
And then so we learned that T-cell therapy. I feel so bad for that one guy that he it just threw itself, you know what I mean? Like, he literally got the mutation that rats get. Well, in the world, it hasn't yet been happened to humans. It happened. Yeah.
Take a note on what was the
I want to read the paper.
what was the actual changes that you got?
I don't know. He didn't say.
I saw that it was a chromosome translocation, but I didn't
Oh, really? Poor guy.
Did you write down the natural response to T-cell?
Yeah, but I did
Oh, okay. Selena, what is one thing your group talked about that you thought was important today? What?
Ubiquitination.
Ubiquitination.
Yeah, ubiquitination. And 3 examples of that are P53, MIG, and beta-catenin.
Right. So we're just adding some of that. And what that is an example of is post-translational regulation. So remember, translation is when a ribosome synthesizes a protein. So that's translation. And so anytime we regulate a protein after translation, that's called post-translational regulation. And one example is phosphorylation. Oh, we've seen lots of examples of that. And now a new one for us is this ubiquitination. And ubiquitination doesn't always target a protein for degradation, but it almost always does. And so we can go back to, right, so this is like a topic from an earlier part in our textbook, but we can go to it with our good examples, right? We can say, oh, I have some examples of proteins that get degraded. All proteins get degraded, Some proteins get degraded really fast, and it's a really important part of regulating them, right? All those proteins, P53, MYC, and beta-catenin, that's part of their regulation. That's really important, switching like that. So that's a great example, just to add some to that. And remember, we're talking about the pathway of gene expression that you learned about in introbio, that full pathway, and sometimes the regulation happens at the level of the proteins. Yay, okay? Anna, what's the thing your group talked about?
We talked about how, like, full circle with the SMAD, about how it can also activate the P21 protein.
Yeah, you know, and so I like that, right? Because we want to talk a little bit about myostatin. This is something different and new. It's at the other end of our life cycle, and we're getting old. And interestingly, the cells that make myostatin hormone are our muscle cells. Our muscle cells themselves are like, we're getting kind of tired of this carrying groceries and crap like that. And so we're just going to start making this hormone and start shutting down the capacity to make more. Yeah.
What happens if you inhibit that?
Yeah, there's
the
people do it. People do it because it turns out myostatin is implicated in some either directly or indirectly in some disease states. So people talk about it with regard to muscular dystrophy, which is a muscle wasting disease. In some cases, it's thought to be a direct involvement, like a misregulation of myostatin. In other cases, maybe you could counteract the disease state by suppressing myostatin and helping the body to quit kind of, putting itself, reducing muscle. Yeah. So yeah, there's a lot of work on inhibiting myostatin. Yeah, therapeutically like that. Yep. Alex had one too.
I had a similar question.
Yeah. And so again, myostatin is and it's a good way for us to expand our awareness a little bit, because we were talking about muscle differentiation in our youth and little kids getting bigger and working out, but at the end of our years, we have other kind of actions. And again, we have an example of transcription regulation, and we're molecular biologists, so we're like right on. But then there are these other examples of RNA processing and translation regulation that we'll talk about a little bit that are important in that late, like what myostatin does late, like that. Let's see, and I think somebody else had a question too, maybe. We can also call out a third person. Let's call in Sam. Sam, did you hear anything you thought was important today?
We talked about ubiquitination, and we talked about SMAD and P53 being multiple enhancers on the P21 gene, and that either one of those transcription factors can induce transcription and stop starch.
Yeah, right. And we saw P21, right? That was early when we were young, and we're like, we got some DNA damage, we got to protect ourselves, right? Here, it's this older age thing, and it's this kind of curious thing where we're like, we're shutting down some of this stuff because I don't know why, it's weird, right? Why are you ever like running out of muscles and being like, yeah, I don't know, But that happens in old people, where they're just like, they get injured and they're just not as good at healing, including muscle injuries. Yeah. Anyway, yeah. So, and then we got to wind this up.
I was just going to be a smart aleck and say we also talked about the CAR T-cell therapy and how it
Good
can
job.
go wrong in many different ways.
Friends of Sam. Yep, Friends of Sam. Yeah. And so we'll talk a little bit more about that. Remember, we kind of jumped ahead into this, but what we want to do is we want to add now some to that, the stories of that pathway, just to add some strength in our knowledge there. And that'll take us to the end of our course, you'll see, because we run into some other kind of tools, removal tools and stuff. All right, great. So keep working on your term papers, and when you're done, let me know.
Can the proteasome also be in the nucleus or just the cytosol?
Is what?
Is the proteasome in the nucleus too, or just the cytosol?
Yeah, and I think it's weird, but I think there are a whole bunch of them in the nucleus, because when people talk about the structure of proteins inside the nucleus, they often say you can take it to the nucleus. So I don't know why they do. Why is that? I don't know.
Boxing right now.
Yeah.
Okay. See you Thursday. Bye.
So, and I have to put it in my brief. I'll send you Yeah. All right. Yep. Hi.
So for my test, should I record with
Oh, yeah, I have it right here.
Oh, okay.
Yeah.
And then
Yes, if you didn't get your test, I have it here. So, Lydia. And
No, I have one. I just had a separate question.
Yeah. And just had one, and let me just check. Okay, yeah. Yeah, do you have other questions?
yeah, just like about, so as far as the P21 and like SMAD and everything, do you need SMAD and P53 together? Because I thought P53 is needed at like period to activate P21. So is it like they kind of build off of each other, or you can just have SMAD?
So you can have one or the other.
Okay.
Is the our
But
understanding.
only when it comes to the
Myostatin, yes.
I wanted to
Yeah,
say
yeah.
Jen, but it must
Yeah,
be okay.
and so PT3 is going to activate P21 in response to DNA damage.
Okay, that's what I
Right,
thought.
and we got to repair that, or else, right? And we would think of that as being in our younger years, where we're like, we're taking care of ourselves. And myostatin is, I mean, for a healthy person, it's an old age story.
Yeah.
And in this old age, they're like, why don't we just activate that P21
Okay.
and just make sure we don't
So
make
it's like
any more muscle.
Just bypassing everything, and it's not about damage, it's
Yeah,
just about
right.
shutting down.
Right, because that's like an existing situation where it's like, like, I don't know, why does that exist, right?
Yeah.
Why do people grow old and
Yeah.
get weak muscles, But that's the important mechanism.
And then, like, I guess follow up, is there any like specific thing that happens that activates like your body being like, okay, we're old, let's just stop, or like it's just sort of like
Yeah, I don't know.
a guessing game?
Yeah, I don't know. I don't like know too much about aging research, because it's so kind of gloomy. Because
Yeah,
I'm like,
it is.
I want to get old and just worn out.
Yeah.
But myostatin, there's also disease states. So like a young
Yeah.
person can have issues with myostatin if it's a
Yeah.
problem, But normally our body doesn't use this myostatin until an older age.
Okay.
When we're like, no, we're done. We don't need to have these stem cells go through the cell cycle and make more muscles. Yeah. Yep. Yes.
I just want to talk about like exam grades and
Yep.
stuff.
Yep.
I haven't done the greatest on either of the exams, but I've like put a lot of effort into studying, and I've realized that just like my biggest issue is how many questions compared to time, and I'm a slow writer, so
Okay.
for me I feel like it doesn't give me a lot of time to like actually think about an answer. I just have
Okay.
to like write things down.
Yep. Yeah.
I was just like, how do I not get
Well,
a
one
poor
thing
grade?
is that the final exam is going to have, all the
Yeah,
time
same
a
length
person
but more
needs.
time, so that will be helpful.
Yeah, and even more than that time. So like if
Yeah.
somebody's like, I just want to work more on it. Yes. Yeah. So, and the final exam is worth more. And,
Right.
and that's built in partly because I really respect it, people can kind of figure stuff out and improve their scores. Yeah. So.
Yeah, because like my grade on this most recent one compared to the first one is higher, but it's still like obviously
Yeah.
not the greatest, and I don't want to.
Mhm. Yeah,
Low
I have
grade.
tremendous respect if you can hang in there and sort it out. And I'm sorry if you felt pressure for time.
Yeah.
We should always talk to instructors about that idea, because it's not supposed to be a race,
Right.
for a grade. But the bottom line is now looking ahead. Yeah, the final exam, you'll have all the time you want, because
Yeah,
I'm
because
not
that's what
going to
I
be in
was
a
like
hurry.
curious,
meeting.
like what is there I can do like help myself?
Well, so what you're doing right now is really, let me just also make sure that.
Because like I've done well in all the